But the final idea I want to throw out here, and what I kind of tried to shape this whole talk around, is the horizontal transfer of genomic segments. Now, in these clonal pathogens, we know that the core genome is absolutely acting as a clone. You know, there's nothing really coming into these genomes to alter them. But perhaps something else is happening. Perhaps there are recombinational elements that are moving out of these large populations back into the microbiome. This is really force. So, in other words, is there something that we might want to think about as evolutionary inertia? So that when you have 10 to the 15th Yersinia pestis out in a field, does that count for anything, or does that DNA just get consumed by other bacteria that are eating it up? Well, in the process of eating it, they might actually get tagged by some of these. So with very large population sizes, very large amounts of DNA out there in the environment, perhaps this is acting as a source, a unidirectional source for sure, but a unidirectional source of DNA that goes on. And in the end, if you're a species, do you live on exactly the same one generation to the next? Well, not in sexual organisms. So why should we really set a different standard for bacterial clonal organisms? Perhaps it's only a portion of your DNA that gets passed on to the next generation, and that really is immortality. So we have an example of that here, and this is where I tie back together these two things. So in Burkholderia pseudomallei, there is a genomic island that is, in fact, look like it came from a Yersinia-like species. It's a Yersinia-like fimbriate genes. It's about 5 kilobases of DNA. What's interesting about it is it's a polymorphism at the population level. So it's found predominantly in those Burkholderia pseudomallei that have crossed the Wallace line, and much rarer back in the ones that have not, as if there's a source population that obtained this, perhaps from a Yersinia, definitely from a Yersinia-like organism, and it moved down. So we need to, if we wanted to answer that question, obviously we have to look for it in different ways, and one way to look for it would be in the growing mass of microbiome data. So with that, I'll point out 4 people who have actually done this work. David Wagner's the vertebrate biologist who was trying to do meta-population analysis on prairie dogs, and they all started to die. Hal Pearson is one of our hardcore cladists. I can't even get him to look at another analysis tool unless it has parsimony in it. Abishai Twanyo is very interested in melioidosis, and Amy Vogler has done a lot of the Yersinia pestis. With that, if I have time, I'll be glad to answer some questions.